All right, hello everybody. Today we're going to talk about Chapter 9, which is Meiosis and the genetic basis of sexual reproduction. Last time we discussed Chapter 8, which was cellular reproduction, and essentially in that chapter we talked about the cell cycle and how cells reproduce via mitosis. Now this week we're going to study meiosis, so a little bit different in the way the terms sound, but they are very different processes. Okay, so 9.1 in our text starts off with an overview of what meiosis is. And the process of meiosis is a type of cell division that reduces chromosome number. That's very important. Meiosis is a reductive division. A reductive division, meaning it reduces the chromosome number within a cell. Also, meiosis functions to shuffle chromosomes in a cell to produce gametes that are genetically different. Okay, genetically different. So if you recall, in mitosis, we had a cell, our first cell here, that divided to become two cells that were genetically identical. That was mitosis. That was last chapter. This time, we're reducing the chromosome number in meiosis and creating gametes, which are genetically different. Part of the process of understanding this chapter's material is understanding what homologous chromosomes are. So we're going to cover that in the overview as well. So homologous chromosomes are members of a chromosome pair. And you can kind of remember what these homologs are and what it means by breaking down your word. So we see homo as our prefix. Homo means same. It's a Latin prefix for the word same. So homologous chromosomes have the same size, they have the same shape, and they have the same construction. And what I mean when I say construction is the location of the centromere. And if you recall, our centromere is what holds together, ooh, that's really poorly drawn, holds together our sister chromatids, which would be right here in the center. Another thing you need to know about homologous chromosomes is they have the same genes for the same traits in the same location. Okay, so homo means same. Homologous chromosomes have the same size, same shape, same construction, same genes for the same traits. Same. Okay, so let's do a little recalling of chromosome structure just for a second. We talked about this already in the past chapter, but we discussed how the DNA in the nucleus contains about 3 billion base pairs worth of DNA in humans, and it's loosely coiled in the nucleus just like this, and we call it chromatin. But when it's tightly coiled down, we call it the chromosome. All right, so this is the structure of a replicated chromosome, actually, because if you recall, chromosomes have linear bar shapes. And then during the cell cycle, in the S phase of interphase, they have a replication process of that DNA that causes them to look like these X's. And they're held together at the center here called the centromere. Right? Now, that also means that these two are genetically identical. So I'm going to put gen ident for short. And therefore, they are called sister chromatids. All right, so all that we talked about last time. All right, so here's some new information I wanted to share with y'all. So something you may not know is that different organisms or different species have different numbers of chromosomes. So some species of fungi, for example, yeast, this particular species of yeast is called Saccharomyces cerevisiae, but they have 32 chromosomes. The garden pea, Piscium sativum, has 14 chromosomes. This species of a fern, which is a plant, has 1,320 chromosomes. The fruit fly, which scientific name is Drosophila melanogaster, has only 8 chromosomes. And Homo sapiens, which are humans, have 46 chromosomes. So my purpose in showing you this is to show you that different species of organisms have different numbers of chromosomes. But what I want you to know from this is humans. Humans have 46 chromosomes in our cells. So every single one of your cells, all of your body cells, except for sperm if you're a male, or eggs if you are a female, all of your other cells other than the sex cells have 46 chromosomes. So previously, when we discussed the last chapter and we talked about cell division, we talked about cells that had chromosome numbers of maybe like 2, right? And then after replication, they look like the X's. All right, but in reality, that's for a organism that has a chromosome number that is very small. Now, in reality, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, and I'm running out of room here. So we can imagine, well, you can see where I'm getting at here. I would have to draw a total of 46 chromosomes in order to give you a representation of what an individual human cell looked like, but I'm out of room, so, but in reality, 46 chromosomes would be in here if I had enough room to draw them. So that just goes to show you how small this DNA is. It's very tightly compacted into this little tiny cell that we can't even see with the human eye. Okay, another thing to know about chromosomes is that they come in pairs, those homologous pairs. So we said human body cells, which we call somatic cells, by the way, that's another term you should know. Somatic cells are body cells. There's all of the cells of your body except sperm and egg, which we call the sex cells, right? So know that definition. Somatic cells are all of the body cells, which is everything except sex cells, which is sperm and egg. Another name for sex cells is gametes. All of that means the same thing. So all of our somatic cells have a total of 46 chromosomes, which means there's 23 pairs, because if you take 46 and divide it by 2, you get 23. Now we can see the arrangement of these chromosomes using a karyotype. A karyotype is another definition you need to know. A karyotype is a picture that shows the arrangement of the homologous chromosomes. So it's basically like if you took a picture of all the chromosomes within the cell and lined them up next to each other in their appropriate pairs. And so if you did that with a human karyotype, you would see 23 pairs. So if we count these up, there's 1, 2, 3 pairs, there's 4 or 5 pairs, and all the way down to 22 pairs, and then this is our last pair. So in humans, you're either going to have this arrangement XX or this arrangement XY for the last set of chromosomes. Okay, so again, 1 through 22, and then this or this will be pair number 23 in humans. Now, something else you need to know is that chromosome pairs 1 through 22 are called autosomes. And the last set of chromosomes are called sex chromosomes. So the last set here is called the sex chromosomes because, well, obviously the name gives it away. They determine sex, male versus female. Whereas the other chromosomes, our autosomes, determine all of our other traits. Essentially, all of these chromosomes contain genes, and genes are segments of DNA that code for a trait like eye color or hair color or height or the shape of your hemoglobin protein or the growth factor concentrations that you produce over your lifetime or gender. Okay, so they control everything. Again, DNA is like a blueprint to tell your cells how to function. Okay, so humans, again, have a total of 46 that are divided up into pairs, which makes 23 pairs. The first 22 pairs are called autosomes, and the last pair are called the sex chromosomes. Now, we said the sex chromosomes, you can have either that configuration, XX, or XY. An XX individual is considered female. So if you are a female, then you have two X chromosomes. Likewise, if you are a male, you will have an X chromosome and a Y chromosome. And each one of these pairs are homologous. And again, as a reminder of what homologous means, homologous, homo means same. They have the same length, as you notice, see the chromosome pair number 1, both of them have the same length. Chromosome pair number 3, they both have the same length. Okay, they have the same order of genes in the exact same location. They're the same size, etc. Another term you need to be familiar with is locus. Locus is a location on a chromosome. Loc literally means location, or the root word, or the root word there is derives from Latin, just like a lot of these do, which means location. Okay, so what that means is that, for example, let's see, this is hypothetical. Let's say that the eye color gene is located right here in humans. Well, if that's the case, then the eye color gene will be located right here on the other homologous chromosome. Let's say, for all intents and purposes, again, this is hypothetical, let's say that the gene for height is located here on that chromosome. Well, then the gene for height is located here on this chromosome as well, and that applies to everyone. If the gene for height is located down here on chromosome 1 for me, it's going to be located there for everyone. All right? Now, something you also need to be familiar with when it comes to homologous chromosomes is that where they come from. One came from mom and one came from dad. And I understand, before I move forward, because I know you're probably looking at this picture right now, is I want you to disregard this stuff right here, because we're not going to cover these terms until a later date. But I really like this picture and the other items that are on it to elaborate what I'm about to explain to you. But so this part, just ignore it for now, because we will learn about what that means later. But anyway, these homologous chromosomes, they come from your parents. When you were conceived, or when I was conceived, or any of us, for every single one of those homologous pairs, one of them came from mom and one of them came from dad. So we have what we call a paternal homolog. That means it came from dad. And a maternal homolog, which means it came from mom. All right. Literally in Latin, pater means father. Mater means mother. I took 4 years of Latin in high school, so I still remember some of this stuff, luckily. But anyway, so again, that's why it's called that, because paternal means it stems from dad, and maternal from mom. Okay, so if we look at the genes that are located along this chromosome, or these chromosomes, we will see that they are located synonymously with the other chromosome. And so what I mean by that is that these genes, which are labeled here as letters and colors, are representing particular traits. And they're the same traits, regardless if it's the maternal or the paternal chromosome. So for example, this gene here, which is labeled in R, which again, remember, this is just a long sequence of A's, T's, G's, and C's, is going to be the same gene that's located here. These are also called loci, okay? Loci, locus, Again, it's just locus is singular, loci is plural. If I'm referring to this locus, I say locus. If I'm referring to both of these locations, I would say loci. But these loci contain genes for the same trait. Again, this hypothetically could be hair color, eye color, the shape of your hemoglobin protein, or whether or not you can taste PTC, which is a chemical that some of us can taste, and it's very bitter, and some of us can't. Okay, so now there can also be minor variations in these genes. So, for example, while both of these genes, which are labeled in little r, lowercase r, can, let's say, again, hypothetically code for eye color, that doesn't mean it's the same color. This could be a blue-eyed allele, and this could be an allele for brown eyes. And that's what variations of genes are referred to as, alleles. So gene 1 if it's could be blue eyes, gene 2 could be brown eyes. We refer to those variations as alleles. But again, these genes are homologous because they are coding for the same thing, which is eye color. It's just the specifics might be slightly variable. Maybe this one down here, C, is for flower color, in plants. Or it could be height. Again, it doesn't really matter, but you can see they're located in the same spot, at the same loci, locations, and therefore they code for the same stuff. But there might be variations in what the stuff is, and we call those alleles. Okay, so let's look at our genome structure in somatic cells versus gametic cells, which are the sex cells. So if we're to take a picture, a karyotype of a somatic cell, in our somatic cells, again, our body cells, everything but the gametes, they have a configuration called diploid. So I want you to look at that word, diploid. Literally, di as a prefix means two. All right, so a way we'll shorthand that is by writing 2N. All right, this means it has a full set of chromosomes. So you can see in every single chromosome number type, we have 2 of them. There's 2 chromosome number 1s, there's 2 chromosome number 2s, 2 chromosome number 3s, and so on and so forth. Each one has a pair. That is a diploid configuration. All right, so in the next picture, we have a haploid configuration. This type of configuration is seen in gametes, which are the sex cells, which are sperm and egg. We say that they are haploid because they have half the number of chromosomes. All right, so here we have, again, diploid. There's 2 chromosomes of each type. In haploid, we see half of that, 1 chromosome of each type. And this is seen in sex cells. Now, why? Why do sperm and egg have a haploid configuration? I want you to think about it. I'll give you just maybe 30 seconds. Okay, so let's think about this. If we had diploid sperm and egg, here would be our egg, and it would have 46 chromosomes. If we had a sperm cell, little tail, what would the resulting offspring's chromosome number be? 92. Is that the chromosome number that I said humans are supposed to have? And the answer is no. We're supposed to have 46 chromosomes. If we have too many or too few chromosomes, it leads to detrimental consequences to the organism. As a matter of fact, this would not even be compatible with life. And most additions or deletion, but a lot of these chromosomal abnormalities in general are just not compatible with life. In fact, about 1 in 4 pregnancies result in miscarriage, and usually that miscarriage occurs before the female even knows she's pregnant, and usually that is because or usually the miscarriage is because there is a chromosomal abnormality, and for the vast majority of the time they're just not compatible with life. Okay, so let's look at haploid cells. So if our egg has 23 chromosomes and our sperm has 23 chromosomes, then our result of the fusion of sperm and egg results in a new organism that has a total of 46 chromosomes. And that's exactly what the cell needs to have to be compatible with life. So that is why sex cells like sperm and egg have a haploid configuration, whereas all of our other cells have a diploid configuration. Okay, and here is another example of a karyotype. So I'm going to back up for just a second. This was an example of a human karyotype that was just an illustration. And this is one that is that was actually a picture that was taken. So you can see that the chromosomes are in lots of different colors. They're basically stained to show different things that the scientist wants to see. But we won't go into the different staining methods in this class. That'd be more of a human genetics course, but it is interesting. But you can see all 22 pairs as well as the last pair, the 23rd pair. And this person is a male, all right? Now, the next topic in our text are human life cycles, and what a life cycle is it refers to all the reproductive events that occur from one generation to the next. So from the time you are conceived till the time that you reproduce. And this process involves both mitosis and meiosis. Mitosis is involved in continued growth of a child. So again, I think I've mentioned this before, but after sperm and egg meet, they create a zygote. You need to know that term. The zygote, by definition, is the first cell of a new individual. So when a baby is made, sperm meets egg, sperm donates its DNA to the egg, and now we got our first cell of our new individual that we call a zygote. That zygote will then perform mitosis many times. And eventually, as mitosis occurs, of course, the zygote is gaining more and more cells. Eventually, there's trillions of cells that make up an adult. But the zygote starts off as a single cell. As more and more cells are created, we form an embryo, and then as more cells are accumulated, it forms a fetus, and the fetus becomes a baby, and then the baby is born, and the baby grows into a toddler, and then a child, and then a preteen, and then a teenager, and then eventually one day an adult. All right, and that's all by gaining more and more cells via the process of mitosis. Okay, and also repair. If we get injured, our bodies repair itself through the process of mitosis. And so because we are doing mitosis, these body cells are all diploid because this fusion of sperm and egg resulted in a 46 chromosomed zygote, and mitosis results in identical daughter cells. We are gaining more and more diploid somatic body cells as mitosis occurs. Okay, so mitosis was last time. But the life cycle involves mitosis because it's the vast majority of what occurs. But meiosis occurs as well during the life cycle of an adult or of a human. Meiosis is a process, though, that reduces the chromosome number from diploid to haploid, and therefore it's how we produce gametes, which are sperm and egg. They only have one of each homologous pair, just like I showed you in the karyotypes. They only had one chromosome number 1 instead of 2. Now, the processes that occur to create these gametes are called spermatogenesis. Break that down. Spermato and genesis. Spermato means relating to sperm, and genesis means the creation of something. So we're creating sperm. This occurs in the testes. So this is where meiosis occurs in the testes. And we call that spermatogenesis because we're creating sperm in men. In females, the process of meiosis is called oogenesis, which produces eggs in the ovaries. And then after eggs and sperm are created via the process of meiosis in the testes and the ovaries, respectively, the sperm and egg can join during intercourse. Dang it. I don't know what I did. How did I do that? Okay. Oh, I just realized I can do captions. That's neat. Cool. I'm going to see how well that works because I think that's pretty interesting. All right, so again, spermatogenesis produces sperm in the testes, while oogenesis produces eggs in the ovaries, and then those eggs and sperm can form a diploid zygote. Okay, so this picture illustrates this human life cycle. So here we will start. We'll start at the zygote. So the zygote was created by fertilization of an egg by a sperm. And then after many, many, many rounds of mitosis, we have a baby. That baby will continuously perform mitosis, and depending on if it is a male baby or a female baby, it will form an adult male or an adult female. Okay, then in the female, in meiosis, her ovaries will produce eggs. And in the male, in his testes, meiosis will produce sperm. The sperm and the egg will fuse. This is a process called fertilization to form a zygote. And then that zygote will perform many rounds of mitosis to create the baby, which will grow up to be an adult. And likewise, the process will continue over and over again, over many generations. Okay. So, how does this process happen? We called it meiosis. Now we're going to go into the details of how meiosis happens, and it's going to look a little bit similar to mitosis, but with some pretty significant differences. Overall, meiosis is a special type of cell division used for sexual reproduction. Remember, mitosis was for asexual, occurring without a partner. Here we have sexual reproduction. Our diploid gamete producing cells in meiosis will undergo 2 rounds of division. We call them meiosis 1 and 2, and ultimately produce 4 haploid gametes. So as a reminder again, in mitosis, we had a cell that divided to form 2 new cells that were genetically identical. That's not what's going on in meiosis. In meiosis, we have a diploid cell that will perform DNA replication, just like in mitosis, but now, instead of producing just 2 identical cells, we're going to have 2 rounds of division, meiosis 1 and then meiosis 2. And that will result in 4 haploid daughter cells. And these are either going to be sperm or egg, depending on if it occurs in male or females. So overall, what happens in meiosis 1 is those homologous pairs of chromosomes will line up, and this is called synapsis. It results in a tetrad, and the reason for that is when they line up, and you'll see this in just a second. We're going to have 4 chromatids per homologous set. These homologous chromosomes of each pair are going to separate in meiosis 1. You need to know that. In meiosis 1, homologous chromosomes separate. In meiosis 2, there's no duplication of chromosomes like there was before the first round of meiosis. So chromosomes are considered a dyad. They have 2 sister chromatids. Di meaning 2. I can't figure out how to erase that still. Oh well. Dyad. The sister chromatids are going to separate. This is just like mitosis. And we have 2 daughter nuclei that will separate. Lastly, the gametes can fuse in the process of fertilization to restore the diploid number and go on to create, give the ingredients to the next generation. Okay, so let's do, what I'm going to do now is talk about the same processes, but we're going to go in a little bit in more detail. And then we're going to do that same thing in even more detail, just to try to, and I'm doing that in this way because I feel like it's going to be the easiest to help you wrap your head around everything that's going on here, because it is a lot. This is a very complex process. So, but in meiosis 1, this is the first round of meiotic division. The chromosomes are replicated prior to its starting of meiosis 1 in interphase, just like mitosis. Remember? Just like mitosis. In mitosis, or before mitosis occurred, which is division of the nuclear contents in cellular reproduction, we duplicated our DNA in the S phase of interphase. Same thing in meiosis 1. So, as a result, each chromosome consists of 2 identical sister chromatids, just like mitosis. Now, this next point is unlike mitosis. So in meiosis 1, homologous chromosomes will pair up to form tetrads. And they're called tetrads because tetra means 4. All right, and let me back up for just a second. See if oh, we mentioned that here. Tetra means 4. 1, 2, 3, 4. So this shows you that again. All right, so here we have our diploid cell. You can see the diploid number is 4. There's 4 chromosomes in that. That means that there's 2 homologous pairs. And it shows you which ones came from mom and which ones came from dad. Dad is in blue. Mom is in red. So you can see this one here would have come from mom. This is the homologous pair for that one that came from dad. And likewise, the shorter ones go together. Because remember, homologous pairs are the same length. They contain the same genes. So after DNA replication occurs, we now have X shapes instead of linear bars. And each one of these X shapes are made up of sister chromatids. They're genetically identical. So what happens in meiosis 1 is that these homologous chromosomes synapse and then separate in such a way that these guys over here are going to move this way and go into this cell. And these two go into that cell. All right, so we divided up our homologous pairs. This is a homologous pair. The blue one went to this cell and the red one went to that cell. Here we have another homologous pair. One of them went to this cell, the other went to that cell. And this is what we're left with after the end of meiosis 1. 2 cells that have one of each homologous chromosome within them. So, because of that, we just reduced the chromosome number. We started off with a diploid number of 4. Now we have haploid cells. They have half the amount of chromosomes as our original cell did. So the next thing that's going to happen is meiosis 2. I forgot I had this slide in here. So this is meiosis 1 continued. So it's just elaborating on a few more things that go on in meiosis 1. But essentially, it's a lot of the similar things that happen in mitosis, except for we're referring to homologous pairs. So in mitosis, we said sister chromatids align on the metaphase plate. Well, this time it's the homologous pair. So we can see them lining up here. This is the same picture from the last slide, and they will be dragged to either pole of the cell, and it will divide just like in mitosis. So those processes of, the fact that it's dividing is the same as what was in mitosis, but it's different stuff that's being divvied up between these two new cells. So our two members of our homologous pair separate in such a way that each of these cells that are created now have a chromosome number of N, which is haploid, whereas the original was diploid, 2N. So now we have N equals 2. Okay, now, after meiosis 1 is complete, there is a short little phase in between meiosis 1 and meiosis 2 that's called interkinesis. And during this phase, the cell's just getting prepared to divide again. There is no more DNA replication. DNA is not replicated again. Now, in meiosis 2, the sister chromatids, so this is where we're at, these sister chromatids are going to move to opposite poles of our cells and divide to create a total of 4 daughter cells that have one sister chromatid apiece. These daughter cells are still considered haploid. Now, the reason for that is because this, meiosis 2, is not a reductive division. Now, you might think, well, we reduced the amount of DNA because here we see sister chromatids, but now we're only seeing half of that in each cell. But the reason why this is not considered reductive division is because, remember, these sister chromatids here and here are genetically identical. So by by separating these sister chromatids into two new cells, we're not altering the DNA that's in the cells because they still have an extra copy. So this is not considered a reductive division like it was in meiosis 1. Okay, so overall, this is where we're going to get into the nitty-gritty now. This is what the cycles of meiosis looks like in detail. So we have interphase in the beginning, just like we did in mitosis. We said we have meiosis 1, and then we also have meiosis 2. Well, meiosis 1 and 2 have subphases. The phases are PMAT, just like you saw in mitosis, except we denote them as prophase 1, metaphase 1, anaphase 1, and telophase 1 in meiosis 1. And then in meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. So what goes on in all of these phases? And a lot of this is going to sound just like mitosis, except for the differences that we already discussed. So in prophase 1, your spindle starts to form out of your centrioles, which are located in the centrosomes. They're moving to opposite poles of the cell. The nuclear envelope starts to fragment, which you can see here. The homologous chromosomes are going to pair up. So you see they're unpaired here. Now this long red one and the long blue one are going to pair up. Likewise, the short ones will pair up. This is called a bivalent or a tetrad. So we already mentioned tetrads. There's just another term for that, which is a bivalent. Bivalent means 2 homologs. Tetrad means 4 chromatids. Now, something else that happens during this phase that's super important is called crossing over. This occurs between homologous chromosomes. And it's where these homologous chromosomes can share genetic information between non-sister chromatids. Let's take a better look at that. So here we have a homologous pair that are in synapsis. They're paired up next to one another. These two in red are sister chromatids. They're genetically identical. That resulted from DNA replication that occurred in the S phase of interphase. Same thing here. The ones in blue, they're sisters. These, the blue one and the red one, are non-sister chromatid. One came from mom and one came from dad, therefore they can't be genetically identical. So what can occur in prophase 1 is crossing over, where we will have literally a flip-flop exchange of genetic material between these non-crypt sister chromatids. So now, if it happens, you can see there's a chunk of the maternal chromosome that traded places with the paternal chromosome. Now, after meiosis is complete, that means we're going to have chromosomes in 4 different gametes that are different than what they would have been without this crossing over event. If these chromosome crossing over events don't occur, then they're always going to have either a completely blue paternal or completely red maternal chromosome. And this process is important because it helps increase genetic variation, and that's critical to the reproductive success of populations, which we'll get into when we discuss evolution. Okay, so that was prophase 1. Again, you need to know about crossing over, and you need to know that it occurs in prophase 1. In metaphase 1, our homologous pairs are going to line up on the metaphase plate. Now, something you need to know about the way they lined up is that they do so independently of one another. That means they do it at random. And this is called the law of independent assortment. Let's look at that in a little more detail to see what I mean. So this cell that has 3 pairs of homologous chromosomes. See, 1 pair, 2 pairs, 3 pairs. Whenever metaphase 1 occurs in this cell, there's 8 possible chromosome alignments, and 1 of them are shown in each cell. So this is possibility number 1 through 8. And depending on which combination occurs, it will have an effect on what the final gametes look like. So that is another methodology that our cells utilize to increase genetic variation. Okay, so you need to know what independent assortment is, why it's important. Okay, so what it is it's where the chromosomes line up independently of one another or at random. The importance is that it increases genetic variation, and it also occurs in metaphase 1. Those are the 3 things you need to know about it. Next, anaphase 1. This is where the homologous chromosomes are going to move apart. Remember, anaphase, the A means apart. The chromosomes are moving apart. They're separating. They move towards opposite poles. Now note, this is not where sister chromatids separate. You can see the sister chromatids are still attached because we have X shapes and not linear bars. So the homologous pairs separated. Right, so this set went that way, and this set went to the other side. Okay, so because we are moving these homologous pairs, we're reducing the homologous pair numbers, or the homolog number, we call this the reductive division. So at the end of this process in telophase 1, after telophase is complete, we have 2 separate cells that now have a haploid configuration of chromosomes. So in telophase 1, the nuclear envelope reforms, the spindle starts to disappear, etc. So now, after telophase 1, we have interkinesis, which is very similar to mitotic interphase, except it's short and no DNA replication occurs. It's just like, okay, the cells are just getting ready to do it again, to go on to meiosis 2. Now, meiosis 2 looks really similar, if not, you could say identical, to mitosis. So here are the 2 cells that resulted from meiosis 1. In prophase 2, the chromosomes will condense, a spindle will form, the nuclear envelope will fragment, your centrioles will move to opposite poles of the cell. In metaphase 2, the sister chromatids align at the metaphase plate, just like in mitosis. They're no longer in homologous pairs like in meiosis 1. You can also see crossing over has occurred, something that doesn't happen in mitosis. In anaphase, the sister chromatids separate and move apart. In telophase 2 and cytokinesis, we have the separation, complete separation of the cytoplasm. You can see the cleavage furrow has formed here on both sides, and pinched off to form four total cells, four daughter cells, and they have a haploid configuration of their chromosomes. They have an n number of chromosomes, haploid, half of what we originally started with. And again, this is not, meiosis 2 is not, that's my symbol for not, a reductive division. And that is because we simply separated sister chromatids which are identical. We did not reduce chromosome numbers. All right, so let's compare meiosis and mitosis. Meiosis is 2 consecutive nuclear divisions, whereas mitosis was only 1. Meiosis resulted in 4 daughter cells, and those daughter cells were genetically different. Mitosis resulted in 2 cells that were genetically identical and only involved 1 round of division. All right. This picture is 9.6 in your book, and it compares mitosis and meiosis via a picture format. All right, so, but essentially all the same things that we just referred to. So in mitosis, there was just 1 round of division that resulted in 2 cells that are genetically identical. In meiosis, we had meiosis 1 and meiosis 2 that resulted in 4 daughter cells that are genetically different and are haploid. All right, so mitosis, 1 division, 2 daughter cells, genetically identical, 2N. In meiosis, 2 divisions result in 4 daughter cells. They're genetically different. And they have haploid configurations. All right, so a few more things that are different between mitosis and meiosis. This is comparisons between meiosis 1 to mitosis, because remember we said meiosis 2 is pretty much identical to mitosis. During prophase 1 of meiosis, synapsis occurs. That's where we have the pairing up of homologous chromosome pairs. We didn't see that in mitosis because there is no such thing as homologous chromosome pairs in mitosis. They had just sister chromatids. During metaphase 1 of meiosis, the tetrads align at the equator of the cell with the homologs, with the homologous chromosomes, facing opposite spindle poles. Again, that didn't happen in mitosis because there are no homologous chromosomes in mitosis. They only have sister chromatids. All right, and then sister chromatids do not separate during anaphase 1. The homologous chromosomes do. During anaphase of mitosis, the sister chromatids separate. Meiosis 2 compared to mitosis, pretty much they're basically identical processes, except that the chromosome configurations are different. Meaning mitosis, we had diploid cells, and at the end of meiosis 2, we have haploid cells. So this picture demonstrates that same information or similar information comparing mitosis and meiosis, but in picture format, which is the way I learned, so I wanted to include this slide. But this shows you the progression of mitosis and meiosis up at the top, and some general synopsis of the differences. So the process of DNA synthesis occurs in the S phase of interphase in both meiosis and mitosis. Remember, after meiosis 1, we have interkinesis, but there's no DNA replication in interkinesis. Synapsis of homologous chromosomes only occurs in prophase 1 of meiosis, but does not occur in mitosis because, again, there are no homologous chromosomes in mitosis. Crossing over only occurs in prophase 1 of meiosis. No crossing over in mitosis. It literally wouldn't make sense because if we had crossing over between sister chromatids which are genetically identical, then we would yield identical chromosomes anyway. Homologous chromosomes line up at the metaphase plate. This happens in metaphase 1. Doesn't occur in mitosis because, again, there are no homologous chromosome pairs in mitosis. Now, sister chromatids line up at the metaphase plate. That happens in metaphase 2 of meiosis, but in metaphase of happens in metaphase 2 of meiosis and during mitosis of, oh my goodness, y'all. It happens in metaphase 2 of meiosis and occurs in just regular old metaphase in mitosis. And the outcomes of these processes are different. 4 haploid cells at the end of meiosis 2 versus 2 diploid cells at the end of mitosis. Okay. Now, something else to know is that mitosis and meiosis occur at different times. Mitosis is a lot more common. It happens all the time, every day throughout our entire life because its whole purpose is for embryonic or fetal or child growth or tissue repair, even as an adult. Meiosis occurs only at certain times in the reproductive life cycle of organisms and only in specialized tissues. So, for example, in humans, let's see, it depends on if it's in men or women, but in women, parts of meiosis occur even in the fetal stages of development, and then it halts until puberty. And then the cycle will, part of the cycles anyway, will continue. We won't go into the details of that in this class. We'll do that in anatomy and physiology if you take that. For men, however, they do not start doing meiosis within the testes until they reach puberty, and they continue doing that for the rest of their lives. Okay, last section in our text is changes in chromosome number. Now, meiosis almost always proceeds normally, and it has to because recall that chromosomal abnormalities are not compatible with life in a lot of circumstances. There's 2 definitions you need to be familiar with here that are new. Euploidy is when you have the correct number of chromosomes. Aneuploidy is when there is an individual that does not have the correct number of chromosomes. There's a change. And we can visualize these changes on a karyotype, which we've already seen. So here is a picture of a karyotype. So if I were to ask you, is this karyotype normal, what would you tell me? And the answer is yes. If you count up all of these chromosomes, there's 2 in every single, 2 of each type, which is normal. Now, the way that one way anyway, that chromosome numbers can change is through non-disjunction, another term you need to be familiar with. Non-disjunction is the failure of chromosomes to separate. This can happen in either meiosis 1 or meiosis 2. If non-disjunction occurs in meiosis 1, then both members of a pair of homologous chromosomes will go into the same daughter cell, and then the other cell will receive none. If it occurs in meiosis 2, the sister chromatids will fail to separate, and that will result in 2 sister chromatids going into a single daughter cell, and the other one receiving none. So ultimately, it's going to result in either a gain or a loss of chromosomes after the fertilization or the union of sperm and egg. And that will result in either a monosomy or a trisomy, depending on whether or not a chromosome was gained or lost. So a monosomy will be where a chromosome is lost. So the cell that was fertilized was one of those gametes that had a missing chromosome. Likewise, a trisomy will be where there is a gain, okay? And so you can think of these as, if you know, mono means 1, tri means 3. So you're supposed to have 2 chromosomes per type. We have 2 chromosome number 1s, 2 chromosome number 2s. If you have a monosomy, then at one of these different types of chromosomes would be missing 1. If you have a trisomy, there would be an extra. So this karyotype here shows trisomy 21. That means there's a trisomy, there's 3 chromosome 21s. And this is what a karyotype would look like for a person that has Down syndrome. Okay, so again, non-disjunction can occur in either meiosis 1 or meiosis 2. The next 2 pictures I'm going to show you show how that progression will look like. I know I already stated it, but again, I'm a picture person, so I want you to see it. So here we have a homologous pair of chromosomes. Now again, this is a simple version. This is just showing you one homologous pair. In reality, if we are looking at a human cell, I mean, you get the gist. There would be lots of X's in here because there would be 46 pairs. But again, this is for simplicity. So if this was a pair of homologous chromosomes after meiosis 1, if non-disjunction occurs, that means that instead of one of these homologous chromosome pairs going that way and one of them going into this cell, instead of that, which would have been normal, both of these get lassoed into one cell. And that means the other cell receives neither. So after meiosis 2 then, the gametes will have 1 less chromosome than what would have been normal. And in this area over here, after meiosis 2, we now have gametes that will have 1 extra chromosomes. And again, these can be sperm or eggs. But what should have been the case is that each one of these gametes should have had one chromatid in them per cell. But instead, these 2 have 2, which is 1 extra, and these 2 have none, which is 1 less than what should have happened. So now that means that if this sperm, let's say this is hypothetically a sperm, fuses with this egg, I'm going to draw a tail on it so we know it's a sperm. We have 0 chromosomes plus 1, which is what should have been in the sperm. That means that will result in a zygote that only has 1 chromosome. But if things had to progress normally, then this cell and this cell, all of these would have only had a single chromosome, plus they would have received a second chromosome from the egg cell, or from the egg, sperm, to make 2. But instead, if these 2 sperm fuse with an egg, they're only going to result in zygotes that have 1 chromosome. Likewise, if these sperms, draw my tails on them, fuse with an egg cell that has 1 chromosome, they're going to result in zygotes that have 3 chromosomes. 2 from dad over here in the sperm, 1 from the mom. And that's not what should have happened. They should have all had 1 apiece. Okay, so if non-disjunction occurs in meiosis 2, that's what this is representing. So we're going back to the same homologous pair we had before. And now this time, we have normal meiosis 1. One of the homologous pairs goes into one cell. The other homologous pair goes into the other cell. Now, if meiosis 2 progresses normally, then one of these sister chromatids will go into cell 1, and the other sister chromatid will go into cell 2, and then we will have normal gametes. If non-disjunction occurs in meiosis 2, then instead of one of these sister chromatids going into the 2 newly forming cells apiece, both of them will be lassoed into the same cell. And that leaves this cell here receiving 1 less and this one receiving 1 more than what would be normal. So if this particular gamete, we'll say it's an egg this time, fuse with a normal sperm, so this is 0. I put -1 because it's 1 less than it should have. And that's going to yield a zygote that only has 1 chromosome. If this sperm, or if we'll say this is an egg, like I said, it doesn't matter. If this is an egg and it fused with a sperm that has that proceeded correctly, it only has 1 chromosome, then this one will yield a zygote that has 3 chromosomes. But if everything happened normally and it fused with a sperm that also has a normal configuration of chromosomes, we will have a zygote that has 2 chromosomes. And then same thing here. All right. So now why did this happen though? Well, I just basically told you that this one got lassoed into, both these sister chromatids got lassoed into the same cell. Why? The reason for that is because, here's my centrioles, and I'm going to draw a single chromosome for simplicity. But remember, we have these little structures called kinetochores on either side of each chromosome. And the spindle fibers are going to reach out and grab that kinetochore, and then they're going to pull on it. The centromere separates and allows for this chromosome or chromatid to be moved to the right side and this one to be moved to the left. Well, in non-disjunction, what happens instead is that kinetochore, centriole, is that spindle fiber doesn't ever make it to the kinetochore. So now, in this case, this entire chromosome, both sister chromatids, are going to be moved to the left, and nothing will be moved to the right, because that's a failure of chromosomes to separate, because the spindle fibers are not connecting properly. And remember, that is one of the things in the cell cycle, one of those checkpoints we're supposed to check off that can sometimes fail. Okay, and so we mentioned Down syndrome already, where it is a trisomy of the 21st chromosome. There are some recognizable characteristics that are telltale signs of Down syndrome. Typically, they're going to have a short stature. They have this eyelid fold. Their fingers are relatively short, and they're typically going to have some mental disabilities that can range in severity. But the chance of a woman having a Down syndrome child increases rapidly with age, starting at about 40. So if you've ever been pregnant or and been to your OB as a woman, then you've probably seen all the signs on the walls. I always see them whenever I've gone before, and when I had my son, they usually do testing now for not just this, but lots of other genetic defects, especially if you have some kind of family history of them. They'll do genetic panel screenings during the very early stages of pregnancy. All right, and so this is a karyotype that represents a person that has Down syndrome. So you can see that the 21st chromosome has 3 chromosomes rather than what it should have, which is 2. Okay, now sometimes we can have abnormal sex chromosome numbers. So whenever I said that chromosomal abnormalities are typically not compatible with life, that primarily was referencing to autosomes. Remember, autosomes are chromosome numbers 1 through 22. Our human bodies do not tolerate changes to autosome numbers very well. There's only 3, I think it's 3 autosomal trisomies that will create or produce a living offspring, and all the others will either, an infant will either not live very long, or there will be a stillbirth, which is where the baby is born not alive, or there will be a miscarriage very early on in the pregnancy. However, abnormalities in sex chromosome number is a lot more tolerable to the human body. So newborns that have abnormal sex chromosomes are much more likely to survive. There's lots of different examples of abnormalities in sex chromosome number. One example is where an individual will have 45 total chromosomes instead of 46 because they're missing one of them. So, for example, if a person has a single X chromosome in their sex chromosome set, but that's it, when they should have an X or another X or a Y, then we call that Turner syndrome. That's where we have an absence of a second sex chromosome. These people are considered technically female. They have a lot of female characteristics, but they do have some telltale. Give me 1 second. My son is screaming at me. Okay, sorry about that. So Turner syndrome is where we have an absence of a second sex chromosome. These individuals only have a single X chromosome. They're considered female because they do not have a Y chromosome. Klinefelter syndrome. These individuals have 47 chromosomes. They have 2 X's and a Y chromosome. They are considered male because they have a Y chromosome, and that extra X chromosome winds up becoming an inactivated bar body. Basically, it coils up really, really tightly, but there's still going to be side effects despite the fact that it is inactivated. So these 2 pictures show you some examples, the 2 examples that I've listed of these abnormal sex chromosome number disorders. The one on the left is a female that has Turner syndrome, and on the right we have a male with Klinefelter syndrome. Some telltale signs of a female with Turner syndrome is obviously the absence of a Y chromosome, but in addition to that, they typically have a webbed neck. So you can see it has a lot more tissue here than what a normal individual would have. They have less breast development because the X chromosome is responsible for the secondary sex characteristics of females, including breast development. So because they have one less X chromosome, that means they have less genes coding for the hormones that cause breast development and therefore smaller breasts. They also, for that same reason, have less developed ovaries. Now, Klinefelter syndrome. These individuals have some breast development. The reason for that is because they have 2 X chromosomes. And again, X chromosomes are going to code for genes that are associated with secondary sex characteristics in females. And so these individuals have more than what is considered normal for a male, and therefore causes them to have some secondary sex characteristics that females typically have, such as the breast development. Also, no facial hair for that same reason. They also have less developed testes because they have higher female hormones than a normal male would. It's all a balance of how much testosterone and how much female hormones they may have. Okay, in addition to that, they also typically have very long arms and very long legs. Okay, another thing that I thought was interesting that I wanted to share that I didn't put in this PowerPoint was that there are other sex chromosome number abnormalities that are similar to Klinefelter syndrome, such as XXXY or even XXXXY. And I thought it was interesting to share that the more X's that are added to this genome, the more severe the side effects are. Typically, starting with this XXXY, you start to see more disabilities, especially cognitive disabilities, and it gets even worse as we see additional X chromosomes. All right, I think that's everything I wanted to discuss for this chapter, so I hope y'all enjoyed the narration of this PowerPoint, and I hope it's helpful to y'all.